sorry about that. This is where we were in the last one, and I got cut off because I got involved in what I was explaining. Um, I wanted to say, and I wanted to pick up where I left off, because we haven't actually even moved from what's happening at the nerve into what's happening in the muscle quite yet. I know I mentioned the sarcolemma, but we're not quite ready to mention the sarcolemma. I guess I jumped the gun a little. The next few slides repeat this information, so if you're feeling like confused, let me just let you know that we do reiterate this information, looking at it from a slightly different angle each time. And all this comes from your book in terms of the pictures. And then the other thing is, I do want to say you do have the Tough Topics, the MP3, that will help you listen to this listen to this again. But the steps of muscle contraction are kind of important. They're important in not only for the class, but for you to understand, because how this muscle contracts and what is needed for a contraction, and understanding it is the key to understanding what drugs do when we actually want to relax, when you prescribe or you give a muscle relaxant, or when you're actually shifting how a muscle works on the patient. So what we did was we got the calcium that is moving along the neuron, and that's causing a change in the permeability, and allowing this neurotransmitter, acetylcholine, to be released, goes in and will lock into receptor sites on the sarcolemma, the junction or the fold of the sarcolemma. That locking in then changes the permeability of the sarcolemma, and that change in permeability causes potassium to be pumped out and sodium to move in to the sarcoplasma. And but more sodium goes in than potassium comes out, which causes this change in polarity and basically creates an action potential. And then this goes on basically as long as the muscle has ATP, but it actually goes on also as long as there's acetylcholine in the synaptic cleft. There is an enzyme that comes down, acetylcholinesterase, that comes down and breaks acetylcholine apart. So there is a time limit on that, on the potential for this contraction. Then this also comes from your book, and basically with neural control number 1, we pick it up where we just left off. So the nerve has released, and you're looking at the top of the picture and also the top of the number sequencing here. The nerve is released neurotransmitters into the synaptic cleft. This is changing the permeability. That permeability is causing calcium to be released from those T tubules. Remember, calcium is stored in the T tubules and at the ends of the sarcoplasmic reticulum, the terminal cisternae, and what happens is that causes a change in the permeability that releases calcium. That is the we're going from the excitation to the release of calcium ions. That release of calcium ions into the area where you have the thin and thick filaments cause the tropomyosin to expose the active sites, the tropomyosin to be shifted, and that exposes the active sites on the thin filaments. So now the thin and thick filaments can attach to each other, and with the presence of ATP, that causes that head of the thick filament to actually bend back, remember, on the hinge area, and that creates the sliding or the shortening of the sarcomere. This will go on as long as there is ATP present. It will go on as long as the chemicals needed to create this contraction will be present, but mostly it's ATP, and when ATP is no longer there to keep the contraction, then the entire process reverses. The active sites are covered, and the thin and thick filaments pull apart, and the muscle goes back to its original state, and that shortening and then relaxation. This is another way to look at it step by step. So you pick your poison, whichever way of looking at it explains it best to you. Look at it and repeat it in your head. This is the actual what's happening between the thick and thin filaments. So you have the thick filament coming down and attaching to the binding sites. That's number one. And then ATP actually activates the head of the thick filament and causes that to push or causes that to pull the thin. So you have the thick pulling the thin toward the M line, and this is going to go on as long as ATP is there, and eventually ATP is going to denature into ADP, but that sets up the process so that the whole thing can happen again. So there's another way to look at it, and look at it even closer with regard to what's happening between the thin and thick filaments and this process of coupling by binding to the active site on the thin filaments, and then that hinged nature of the thick filament of the head being actually pulled back. I guess that, kind of it hinges back, but that actually causes the thin filaments to pull. So it's actually pulling it. You can think it's like pulling a rope by that connection. That causes the sarcomere to shorten. All of this is happening in these each units simultaneously. So as a group, they all shorten, and we consider that a muscle contraction. We see that as a muscle contraction. And then again, third way to look at it, like I said, you pick your poison, whichever one resonates for you. We're just doing it over again here. So you have the acetylcholine release, so the neurotransmitter released into the synaptic cleft. That's coming from the nervous system. That then changes the permeability and causes the T-tubules to release their stored form of calcium. So they're stored calcium ions. So I should say it like that because it sounds kind of like I'm talking about bones. So that calcium ion was when this muscle relaxed, calcium ions were taken up into the T-tubules and basically sitting there waiting for the next contraction. And then that release causes an exposure of the active site and allows the cross bridge formation. That then starts the contraction cycle, the pivoting, the moving of the thin filaments in toward the M line. And that's the steps that initiate a muscle contraction, steps that end the muscle contraction. The acetylcholine is broken down by the enzyme acetylcholinesterase that ends that permeability. Calcium is reabsorbed into the T-tubules, so it goes back into the sarcoplasmic reticulum, and Remember, the sarcoplasmic reticulum is kind of the bluish lace apron-y thing that's around, apron, I shouldn't say thing, the bluish open network that is around, that extends from the Z line to the Z line, and then at the Z line of the sarcomere you have these T tubules. So that calcium is taken back up again and waiting for the next contraction. The active sites are covered. With regard to the thick filaments, the ATP is denatured, and it loses its ability to do work. It turns into ADP, waiting to be able to have a phosphorus put back on again, and that ends the contraction, and then the muscle relaxes. Everything goes back into place. This process happens over and over again in very, very regular patterns. So the patterns are regular, that it's not necessarily the same on every muscle in terms of the time limit, but the relationship of time limits are very similar in every muscle group you have. You have a resting phase, which is which is a very brief phase, and then you have a phase considered the latent phase, where you actually have the ability for something to happen. So something can happen in the latent phase, and if in fact there's a stimulus, you do go into the contraction phase all the way to the maximum tension. So all the way to, and again in your head you're thinking of the thin and thick filaments, you're thinking of those titin proteins and how compressed we can get those thin and thick filaments without going into the zone where thin thick filaments don't exist, and not compressing it to the capacity beyond what that muscle can do, and that maximum tension. And then what's going to happen is the relaxation phase is going to take place when ATP is gone. Calcium gets back, is reloaded back up into the sarcoplasmic reticulum, and the whole muscle is waiting to have another stimulus and to start another contraction. But that isn't possible during the resting phase. You have to wait for the stimulus and the latent phase to allow another contract. Not normally, it's not normally possible. Or it isn't normally, it doesn't normally happen. I don't want to say it's not normally possible, because you do have some abnormal normals. So with regard to different abnormal, what can be normal but also can be a symptom of a disorder in terms of our particular patient, is you have this step, this TREP, that actually can the tension can increase. So you have an increase in the peak of the tension with each successive stimulus, and so that stimulus is right after the relaxation phase, and each one is successively the tension increases. And then you have this idea of a wave summation, where each stimulus will arrive before the relaxation phase is totally complete. So you actually don't have a full cycle of relaxation phase. And like I said, this it's possible this can happen it's generally, I guess I just shouldn't wade into the normal, non-normal. You can have varieties of both of these problems, where you can have problems, you can have the problems in a normal patient, or you can have a variety of these problems in somebody who has certain musculoskeletal disorders. Incomplete tetanus and complete tetanus. With incomplete tetanus, you do have some relaxation, but it's very brief. And with complete tetanus, there is no relaxation. you can see where without relaxation, what you're constantly, I'll get you next time.